Hi everyone and welcome to the internet. What we're going to be doing in today's lesson is having a look at a process called the transpiration stream. So what we're going to hopefully be able to do by the end of today's lesson is to explain what transpiration is and how the transpiration stream actually happens. To start us off though, we've got 3 questions based around our previous lessons, so have a go at answering those 3 questions in your book. So what I'm going to do is give you about 4 minutes to have a go at that. Let's make sure we've got the right answers here then. So the 2 types of transport tissue in plants are xylem and phloem. Give yourself a mark for each of those. Question 2, those are contained within structures called the vascular bundles. Give yourself a tick for that one. And then to compare our xylem and phloem, then the key thing here is that when you're asked to compare, you give something for xylem and something for phloem. So you can't just say xylem's made of dead cells full stop and then expect to get good marks on these questions. So you'd have to talk about what xylem's made of and what phloem's made of. So let's go through these ones. Xylem is made of dead cells, give yourself a tick for that bit, but phloem is made of living cells. Get that one. Xylem transports water, 1 mark, whereas phloem transports sugars, 2nd mark. Xylem have no end walls, 1 mark, but phloem have sieve plates, 2nd mark. Xylem carries substances from the roots to the leaves, 1 mark, but phloem transports sugars in both directions, 1 mark. So add up your total score. It is out of 11 this time. So add up your scores out of 11 and write those in your book. To start us off, what we need to know is this definition. Now the good news is that if you learn this definition as I've given it to you on the screen, then it's going to hold good when you do A-level biology as well. So it's the same definition for GCSE and A-level. So transpiration is the loss of water vapour from the aerial parts of a plant. So you could get 2 marks for this. First one for saying loss of water vapour, and you could phrase that as evaporated water, that's fine. But if you just say loss of water, no, it's not going to get you anything. So loss of water vapour, and the 2nd mark from the aerial parts of a plant, or you could phrase that as from the leaves. So the best definition you could give is the loss of water vapour from the aerial parts of a plant. But if you can remember that water evaporates from the leaves, then you're going to at least have a good shot there. But as I say, this is your textbook definition you should learn. Thinking about what we've done in our previous lesson on those transport tissues in plants, and thinking about the structure of plants and the work we did right back at the beginning of B2, see if you can have a go at explaining how water gets to the leaf. And I want you to start with water in the soil and then give as much detail as you can on how it gets from the soil to the leaf and leaves the leaf. So I'm going to give you about 3 minutes to have a go at this. To start this off, we need to go all the way back to the beginning of B2, where we had a look at the different processes by which substances move. And the one we're going to look at here is osmosis. So our first mark is for saying that water is taken in at the roots, and your second mark associated with that is for saying by osmosis. And hopefully we remember the definition of osmosis is that water is moving from an area of high water concentration to an area of low water concentration across a partially permeable membrane. The next bit we need to talk about is what happens once it's got into the root. So your next mark is for saying that water travels from cell to cell in the root until it reaches the xylem. Then our final mark is for saying that it travels up the xylem to the leaves. So hopefully we all at least got 2 marks in there. I'd hope that we got 3. I think that the water travelling from cell to cell in the roots, maybe that was the tricky one in there. But give yourself a mark out of 4 for that section. If you didn't get all of those, I'd suggest you take a minute now to write down those 3 sentences. What I've given you on the screen then, because we can't do the normal experiment here, then I've given you an image from a microscope. So this image here is showing us the underside of a plant leaf. Now, what you'd normally do if we were in school is you'd have some leaves, you'd get some clear nail varnish and paint a thin layer on the bottom there, or sometimes you'd use sellotape to do this. Clear nail varnish is better in my opinion. And then you place that onto a slide once it's dried and look at it under a microscope and you can see the stomata. So as you're looking at the image I've given you on the screen, hopefully you can make out that there's these little sort of areas that look kind of like eyes, almost like cat's eyes. So you've got a green outside, a yellow inside, and then the black in the middle. So that is the stomata, the black in the middle there. That is the hole that leads from the inside of the leaf to the outside. So what I'd like you to do is, using this microscope image that we've got on the screen, is just draw a little sketch of a small section. You don't have to draw the whole thing, just a small section that has at least one of the stomata in there, and label it for me. So I'll give you a couple of minutes to do that. What we've got in these two pictures then are another really important structure that we find around the stomata. And that structure is called a guard cell. So guard cells come in pairs, as you can see in the pictures there. And what happens is they will either be open or closed for the stomata. So if we have a look at the one on the left first of all, that's where the guard cells are turgid and the stomata opens. Whereas the picture on the right, that's where the stomata are closed because the guard cells are flaccid. Now, the way this works is easiest if you put your hands together, so kind of like, typical prayer position. And then if you imagine that the inside, so your palms, actually have a thicker cellulose wall. And you can see it in the picture there, it's probably easier on the one on the left. So we've got a thicker wall on our palms than on the outside of our hands. Now, what's going to happen is, as water moves into the guard cells, it goes into the vacuole swells and pushes out against the cell wall, then it's actually going to end up pushing against the outer part of the cell more, because it's a thinner wall than the inside. So that means the outside is actually going to start to push outwards more than the inside. So with your hands that are in your prayer position, if you then move so your fingertips are pushing against each other, so have your palm, the very heel of your hand, and your fingertips push against each other, then what you should find is that you kind of almost form an arch with each hand and you've got a gap in the middle. That's exactly what's happening with our guard cells. So as water moves into the vacuole, it swells and pushes out against the cell wall, and because we've got a thinner cell wall on the outside than the inside of our guard cells, they push outwards and open the stomata. When we don't have enough water in our plant cells, then what happens is the guard cells are going to become flaccid because the vacuole isn't going to be pushing against the cell wall, and therefore they're going to be closed and the stomata will not allow anything to pass through it. So what I'd like you to do then is have a go at explaining how the guard cells control the stomata, and you might want to draw the diagrams to help with this. So I'm going to give you about 4 minutes to have a go at this. So let's double check we've got the right explanation in there. When we've got plenty of water, the vacuole pushes out against the cell wall and the thinner outer wall bulges outwards, which opens the stomata. That's our diagram on the left. When there's limited water, the vacuole doesn't push out against the cell wall, the cell is flaccid and the stomata are closed. And I've misspelt stomata there, so please just correct it. It's not stomati, it should be stomata with an A at the end. And that's the diagram on the right there. So make sure you get those points written down if you didn't get them in your explanation in your own words. So what we've got here then is a little animation just showing us what's happening in the transpiration stream. So the little blue circles that we can see moving through the diagram, those are representing our water molecules. So what we've actually got then is down here, we've got our root hair cell. These are the structures in our roots. This is our xylem vessel moving up the stem. And then up here, this is our leaf. So we've got a cross section of a leaf. Hopefully that looks familiar to you. So what we've got up here are the palisade cells, and we know they're palisade cells because they're packed full of the chloroplasts there. We've got our epidermis on the top. We've then got the spongy mesophyll, these ones here, and the air space. We've got our guard cells here, the stomata in the middle, and then the lower epidermis there. So what we know happens then is that down here, obviously our root is in the soil. So water in the soil is then going to be taken up by the root hair cell through the process of osmosis, and then it's going to move through the root. So if we have a little look at that in a bit more detail, we've got a high water potential in the soil, a lower water potential in our root hair cell, so that the water is going to move in through that process of osmosis. Then it's going to travel through the cell, cell to cell. Don't worry about these names up here, that's if you go on to do A-level biology, you'll need to know that. And then it ends up in the xylem vessel. So once it's in the xylem vessel, it's going to be moving up the actual xylem itself. And what happens is it's then going to have this continuous column of water that's being drawn up. Because what we see is we're losing water out the top, and therefore it draws more water in at the bottom. And again, something you'll go into a lot more detail on if you do A-level biology in the future. So as it travels up the xylem, it's then going through the stem and ends up in the leaf. Now, if we watch what happens up in the leaf here, you can see that one went straight out through the airspace out of the stomata. But they don't all do that. This one has gone up into the palisade cells to be used in the process of photosynthesis in the chloroplasts. So some of the water will actually be used in our plant cells. And what we actually see happening then is the water comes through the xylem into the air spaces, and then it's going to diffuse out of the stomata. Because what we've got is a higher water potential here inside the leaf compared to here outside the leaf. So it's going to move out of our actual leaf by diffusion through the stomata. So now we've seen the animation, what I want you to do is have a go at actually describing the transpiration stream and explaining how the water moves from the soil to the leaf with as much detail as you can. And you can see there the difference between your kind of up to grade 5 and then the over grade 5 answers is if you're just describing it in quite simple terms, you're down at the grade 5 and below. Whereas if you're actually explaining each of those stages using the correct terminology, that's moving you up to the 6, the 7 and beyond. So have a go at actually explaining what's happening in the transpiration stream. I've given you the little picture there for our animation, hopefully to remind you of those key parts. So I'm going to give you about 3 minutes to do that. Okay, this should be your full explanation then. Any of these points you're missing, obviously get them written in your books. If you have got them, give yourself a tick. So the first part is that water enters the plant at the root by osmosis. It moves cell to cell through the root until it reaches the xylem. It travels up the xylem to the leaf. So I'd hope we got those 3 points first of all. The reason that it moves up the xylem is because the pressure at the top of the xylem is lower than the pressure at the bottom. So that means water moves from high pressure to low pressure. Some of the water is then used in the palisade cells in photosynthesis, and some water passes into the air spaces and then out of the stomata by diffusion, and it goes into the air surrounding the leaf. So if you still need any of these points, it's going to be up for 1 more minute, but obviously you can pause this video if you need more time to do that. Now, if we are pretty terrible at keeping plants, and I know there's a very good reason I don't really have houseplants inside my house because I'm pretty rubbish at remembering to water them, then what happens is they will wilt. So what I want you to have a go at doing is answering these two questions in your book. Why do plants wilt, first of all? And what happens when plants have wilted? So I'll give you just about a minute and a half to answer those two questions. So first one, the reason that plants wilt is because they're losing water at a faster rate than they're taking it in. I.e., you forget to water your plant for about a month and it's going to still lose water. So that's going to lead to a pretty sad looking plant. In terms of what happens when it wilts, then the leaves collapse and they start to droop. As the leaves are drooping, then it reduces the surface area available for evaporation. The stomata will close because obviously the guard cells are flaccid, and that means we're no longer able to take in the carbon dioxide from the atmosphere because it moves in through those stomata. So if they're closed, we can't get carbon dioxide going in, therefore photosynthesis will stop. And if this happens over a prolonged period, then it's goodbye to your plant, I'm afraid. So give yourself a tick for any of those you got right. Last thing to do today then is the PreNuri. So those who with target grades up to a grade 5, it's the two columns on the left. Those who with target grades over a grade 5, it's the two columns on the right. So I'm going to leave these up for a couple of minutes today, but if you need longer, obviously hit pause and take as long as you need to do this. As always, you can find the answers to the PreNuri over on my website, sciencewright.weebly.com. You'll also be able to find that under the Internet tab. Just select the week that we're in, and then you'll be able to find things like the lesson PowerPoints, the answers to the PreNuri, any additional work, and if there's any little experiments I thought you could do at home, then they're there too. So have a look and get using that.